We're continuing our discussion of medical genetics. I'm Dr. Reichenbaker, and we're moving on to Chapter 3 in your study book. The topic in Chapter 3 is cytogenetics. In cytogenetics, we study microscopically observable alterations in chromosomes. That is, in cytogenetics, we look at alterations in chromosomes that are so big we can actually see them in the microscope. The kinds of mutations that we talked about earlier for single gene diseases, missense mutations and nonsense mutations and so forth, are much too small to see in a microscope. But in this chapter we'll be talking about big changes in either number or structure of chromosomes that we can actually see in a microscope. These kinds of problems are very, very common, and in fact on this slide it will tell you that 1 in every 150 babies born alive there is some kind of chromosome problem. It's one of the leading causes of mental retardation. 1 in every 150 babies, that's an extremely high number. And that number even is only the tip of the iceberg, because that only represents the babies that are born alive. The vast majority of the time, when there is a fertilized egg with a chromosome problem, that early embryo is lost prenatally, and there's a spontaneous abortion, and this represents the primary cause of loss of pregnancy. So cytogenetic problems, chromosome problems, are extremely common and extremely important for us in medicine. This slide describes chromosomes and the way their appearance in different parts of the cell cycle. As we go through this chapter, we'll see a large number of diagrams that represent chromosomes, and sometimes those chromosomes will be represented in the way the chromosome is represented on the right-hand side of this particular slide. That is 2 arms separated by a constriction in the middle called the centromere. However, sometimes in some drawings the chromosome will be represented as it is on the left-hand side of this slide. That is basically a doublet. That is essentially a double chromosome, again with a constriction in the middle. Well, why do we represent chromosomes in those 2 different ways? It's because of the way chromosomes occur in the cell in different parts of the cell cycle. The circle there in the middle of the screen represents the cell cycle. M refers to mitosis or cell division. G1 refers to the period of time immediately after the cell has divided, but before the cell has replicated its DNA. The S phase represents the period of time for DNA replication or DNA synthesis, and G2 is the period of time after the DNA has been replicated but before the cell divides. Now, in G1, as the cell is preparing for DNA replication, we believe that each individual chromosome contains 1 long linear molecule of DNA. A DNA molecule is not in one long stiff rod. That is, it's all wrapped around itself in 3-dimensional space and bound with histones and other components to form chromatin. But each chromosome contains 1 long linear double-stranded DNA molecule. After the cell goes through the S phase, DNA replication, then we have made a duplicate of that molecule of DNA, and both of those daughter DNA molecules become associated with histones to form chromatin. But those 2 DNA molecules are not completely pulled apart yet. They are still held together at the centromere. And so in the G2 phase, the picture that we have there represents still 1 chromosome, but that 1 single chromosome contains 2 DNA molecules and is made up of 2 sister chromatids. Those 2 sister chromatids then will come apart when the cell goes through mitosis or cell division, bringing us back to G1. So this is the reason that we represent chromosomes in two different ways because of the way those chromosomes look in different parts of the cell cycle. In your book, there are two figures that picture chromosomes for you. At the top of this page in your book, you'll find an artist's drawing of chromosomes. And at the bottom of this page, you will find an actual photograph of real human chromosomes. The artist at the top, who drew the picture at the top of this page, has drawn just one copy of each chromosome, and those chromosomes are drawn as they would exist as a pair of sister chromatids in the G2 phase of the cell cycle. When we perform a human karyotype, that is, when we look at human chromosomes, we look at those chromosomes in the metaphase stage of cell division. And the reason we do that is because that is the period of time in which the chromosomes are the most highly condensed and most highly visible in the microscope. So these pictures will represent metaphase chromosomes. And in metaphase, each chromosome consists of a pair of sister chromatids. Now, we can distinguish the chromosomes from each other based on several factors. A normal individual contains 23 pairs of chromosomes for a total of 46. The first 22 pairs of chromosomes are called the autosomes. Normal individuals have 2 copies of each of those. The 23rd pair then is the sex chromosomes, and you either have XX if you're female or XY if you're male. The chromosomes can be distinguished from each other based, in one instance, on size. That is, the autosomes are placed in order and numbered in order of decreasing size. So for the autosomes, chromosome number 1 is the largest, chromosome number 22 is the smallest. The chromosomes can also be distinguished from each other by the position of the centromere, the constriction that separates the two arms. That centromere, that constriction, separates each chromosome into a smaller P arm and a longer Q arm. If that centromere is approximately in the middle of the chromosome, then we refer to that as a metacentric chromosome. If the constriction is somewhat closer to one end, as it would be for chromosome number 9 or 10, for example, we refer to those chromosomes as submetacentric chromosomes. And if the centromere is very close to the ends of chromosomes are called telomeres. If the centromere is very close to the telomere, as it is in chromosome number 21, for example, we refer to that as an acrocentric chromosome. So the position of the centromere can be used to distinguish one chromosome from another. We can also distinguish the individual chromosomes by their banding patterns. In this diagram, you will notice a series of black and white and gray bands that are drawn on each chromosome. The pattern of those bands is characteristic for each chromosome, and those bands are artificial. That is, we put those bands on the chromosomes in the laboratory by staining the chromosomes with a series of dyes that will bind more tightly to some chromosome regions than another. So, an expert in cytogenetics can break open a cell, take a picture of the stained chromosomes, and from the size, position of the centromere, and the banding patterns, an expert can distinguish each different chromosome from all the others. The next slide shows you an actual photograph of a real human karyotype showing the 2 copies of each chromosome in a normal individual. This individual would be a male, a normal male, because the person has 2 copies of each of the 22 autosomes, and then an X and a Y chromosome. So a normal individual has 23 pairs, or a total of 46 chromosomes. what can go wrong? What kind of problems can we have with chromosomes? Chromosome abnormalities fall into two basic categories: numerical abnormalities and structural abnormalities. That is, we can have problems with either having an incorrect number of chromosomes, or we can have problems from having an incorrect structure of some chromosomes. Let's look at the numerical abnormalities first. Within the concept of numbers of chromosomes, there are two major terms, euploidy and aneuploidy. Euploidy refers to any cell that has some multiple of 23 chromosomes, 23, 46, 69, or 92 chromosomes. That would be euploidy. Aneuploidy refers to a cell that has any number that is not a multiple of 23. Most commonly either 45 or 47, but technically any number that is not a multiple of 23. Let's look at euploidy first. Now we all have, that is normal individuals have, two perfectly normal kinds of euploid cells. We have our gametes, which are haploid. That is, our gametes, our eggs and our sperm, have one copy of each chromosome for a total of 23. We refer to those gametes as haploid. We also have our somatic cells, that is, our skin cells, liver cells, kidney cells, which have two copies of each chromosome. We refer to those normal cells as diploid. They would have 2 times 23 for a total of 46. Here comes an abnormal euploid cell called a triploid cell. Every once in a while, a single egg can become fertilized by 2 sperm. If that were to happen, then that fertilized egg and the baby, if it goes on to be born, would have 3 copies of each chromosome, 3 times 23 for a total of 69. Each individual cell would be called a triploid cell. The syndrome would be called triploidy. Triploidy, just add a Y to the word triploid. About 1 in every 10,000 babies born alive is an example of triploidy. These babies are very severely affected and do not survive very long. Even more severe is tetraploidy. Tetraploidy, in which a single egg has become fertilized by 3 sperm. In this case, the fertilized egg and the baby, if it is born, would have 4 copies of each chromosome, 4 times 23 for a total of 92 chromosomes. This condition is very rare and is lethal. The babies die very, very quickly after birth. Now, the other kind of numerical abnormality that we can have is called aneuploidy. Aneuploidy refers to a situation in which a cell has the incorrect number of chromosomes, but it's not a multiple of 23. Typically, aneuploid cells result from the loss or gain of particular chromosomes, most commonly the loss or gain of 1 single chromosome. So most examples of aneuploidy would involve an individual who has either 45 or 47 chromosomes, 1 missing or 1 extra. Aneuploidy, as we will see in a few minutes, is most commonly caused by non-disjunction during meiosis. That is, failure of the chromosomes to separate properly from each other during meiosis. We will look in more detail at how that happens in a few minutes. Now, theoretically, on paper, any chromosome could be missing or any chromosome could be extra in a cell, but in reality, the vast majority of those would lead to a loss of pregnancy, spontaneous abortion, and the baby would never be born. That is, most examples of aneuploidy are not compatible with life. So for your exam, we really only need to focus on the ones that are compatible with life, in which where we do have examples of these patients in the clinic, and these are the ones that are most likely to appear on your exam. So let's narrow down the possibilities. On the screen it says all autosomal monosomies are lethal. That simply means that in the clinic you will not have any patients that have a missing autosome. A monosomy means having only one copy of a particular chromosome. So there will be no patients with having a single autosome missing, because all of those examples are result in lost pregnancies. And in fact, most trisomies also result in loss of pregnancy, and the patients are never born. That is, a trisomy means having 3 copies of a particular chromosome. Most of those are not compatible with life. However, there are 3 autosomal trisomies that are compatible with life. These are the ones that are found in the clinic and that you may see on the exam. The trisomies compatible with life are trisomies for chromosomes 13, 18, and 21. The most famous of these is trisomy 21, or Down syndrome. The picture on the left shows an example of a child with Down syndrome. The picture on the right shows a karyotype of such a patient. In the karyotype, you can see that there are 2 copies of each chromosome, except for chromosome number 21, in which there are 3 copies of that particular chromosome. On your exam, you are very likely to be shown a karyotype, and you will be asked to identify the particular syndrome that matches that karyotype. When you're shown such a picture on your exam, you will be able to see the picture very clearly and be able to see the numbering system very clearly. They may not have a big black arrow pointing to the problem as we do in this particular slide, but you should be able to recognize the problem and identify the particular syndrome associated with that karyotype. Here are the names of the 3 autosomal trisomies that you must memorize to prepare for your exam. Trisomy 21 is Down syndrome. Trisomy 18 is Edwards syndrome. Trisomy 13 is Patau syndrome. Now we can also have aneuploidies of the sex chromosomes, and the 2 important aneuploidies of the sex chromosomes are Klinefelter syndrome and Turner syndrome. This slide shows a patient with Klinefelter syndrome. Klinefelter syndrome patients are males with XXY. That is, Klinefelter patients have a total of 47 chromosomes, including XXY. Klinefelter males are usually very tall and thin, and you can see some breast development, for example, in this picture. With regard to sex determination, if you have a Y chromosome, you are a male. If you do not have a Y chromosome, you are female. So Klinefelter patients, since they have a Y chromosome, are phenotypically male. Now, they are not completely normal males. They are sterile, for example, but phenotypically they are male. Klinefelter patients will show inside their cells something that we ordinarily only find in normal females, and that is Barr bodies. Since Klinefelter patients have 2 X chromosomes, early in their development, randomly within each cell, one or the other of those chromosomes is condensed to form a Barr body. There are some individuals that actually have 3 X chromosomes and a Y. Those individuals have characteristics like Klinefelter's patients, but with more severe symptoms. Those patients with 3 X's and a Y would have 2 Barr bodies within each cell, because anytime a cell has more than 1 X chromosome, all the copies but one in each cell are inactivated and condensed to form a Barr body. This slide also shows the karyotype for a Klinefelter's patient, and if you look at that karyotype, you should see 2 copies of each of the autosomes, but down in the lower right corner you will see 2 X's and a Y, Klinefelter syndrome. The other important sex chromosome aneuploidy is known as Turner syndrome. Turner syndrome patients are females with just 1 single X chromosome. So Turner syndrome patients have 45 chromosomes, just 1 single X. In the left-hand part of this slide, you'll see a picture of a Turner syndrome patient. These patients are usually relatively short. They have a webbed neck, that is, there's extra skin in their neck area. It sort of looks like webbing. And another one of their common characteristics is a broad shield-shaped chest with widely spaced nipples. You cannot see all of those characteristics in this particular picture. On the right-hand part of this slide is a karyotype for a Turner syndrome patient, and in this karyotype you should be able to see 2 copies of each chromosome, but just 1 single X. Since Turner syndrome patients do not have a Y chromosome, they are phenotypically female. They are not normal females, but they are phenotypically female. Where do these aneuploidies come from? The aneuploidies most commonly occur because of non-disjunction during meiosis. That means failure of the chromosomes to separate from each other properly during meiosis as we produce our sperm or our eggs. This diagram illustrates normal meiosis, that is, the normal separation of chromosomes during meiosis. The diagram only shows 1 chromosome pair. Of course, in real life, each cell would have 23 pairs of chromosomes. The cell on the extreme left is the starting point, the cell that has 1 copy of each chromosome. The first thing that happens is that cell goes through the S phase, that is DNA replication, and following DNA replication then, each chromosome consists of a pair of sister chromatids. That cell then will undergo 2 meiotic cell divisions that we call meiosis I and meiosis II to separate or segregate those chromosomes from each other. In the first cell division, meiosis I, what happens is that the 2 chromosomes separate from each other. In the second cell division, or meiosis II, the sister chromatids separate from each other. So that the end product over on the right, the gametes, whether we're referring to eggs or sperm, the gametes then will contain just 1 copy of each chromosome for a total of 23 chromosomes in the normal gametes. But we can have non-disjunction occurring in either meiosis I or meiosis II. This diagram shows non-disjunction during meiosis I. And as you should be able to see in the middle of this diagram, in meiosis I, what has happened here is that this particular chromosome pair has not segregated properly in meiosis I. Both copies of the chromosome went to one cell, none went to the other. Meiosis II did occur correctly in these cells, but because of the problem there in meiosis I, when we look at the gametes over on the far right of this slide, we find some gametes who have 1 extra chromosome, and we find some gametes that have a chromosome missing. Now, the result in the child will depend on exactly which chromosome is involved in the non-disjunction and which of those gametes is involved in producing the fertilized egg that leads to the child. The 2 gametes shown at the top over there on the right, have the ones having an extra chromosome, could result in the trisomies, for example, Down syndrome or Edwards or Patau or Klinefelter's syndrome if the extra chromosome is an X chromosome. The two gametes near the bottom on the right that are missing a chromosome could result, for example, in Turner syndrome if that missing chromosome is an X chromosome. We can also have non-disjunction occurring in meiosis II. In this diagram, meiosis I occurred properly. The chromosomes did separate from each other properly in meiosis I, but in meiosis II, the cell nearest the bottom of the screen showed non-disjunction. That is, the sister chromatids did not segregate properly from each other during meiosis II. Now, again, we end up at the bottom right of the screen with a gamete with one extra chromosome and a gamete missing a chromosome. And again, the result in the child will depend on which of those gametes is involved in fertilizing or in forming the fertilized egg. So, an important thing to remember is the aneuploidies are most commonly caused by non-disjunction during meiosis. So, for example, on your exam, if you saw a test question that asked, what is the most common cause of Down syndrome, your answer, the best answer would be non-disjunction during meiosis, and your best answer would also say in an older mother, because non-disjunction tends to be more common in older women. The main reason for that is that a female is born with all of her eggs, and those eggs are locked in an earlier stage of meiosis. And those eggs only undergo the meiotic cell divisions when they are released 20 or 30 or 40 years later. And it turns out that the older a woman gets, the more likely it is that non-disjunction will occur. And this is the reason that we will commonly recommend amniocentesis for older pregnant women. Okay, so for your exam, the best answer of the cause for Down syndrome would be non-disjunction during meiosis in an older mother. Now, older fathers are not off the hook, although we do not believe that non-disjunction is a problem in older fathers, because males continually produce sperm their entire lives. However, we are beginning to find some diseases in which there's an association with having an older father. Schizophrenia is one such example. We believe that the problem in older fathers is an accumulation of mutations. That is, simply as a man lives longer and longer, during his lifetime he accumulates more mutations, and so therefore those mutations can appear in his sperm at an older age, and leading to certain diseases. Now, in addition to the numerical abnormalities of chromosomes, we can also have structural abnormalities of chromosomes. That is, chromosomes that have an incorrect or rearranged structure. As we look through some of the following diagrams, we will see some structural alterations that I will refer to as balanced alterations, and some that I refer to as unbalanced alterations. A balanced alteration is one that results in no net loss or gain of genetic material. That is, sometimes, even though the structure of a chromosome has been arranged in a peculiar way, sometimes the person with those chromosomes will still have 2 copies of all of his genes, which is what a normal individual is supposed to have. Most commonly, a person with such a balanced alteration will still be okay. That is, that person will not have the symptoms of any disease because the person still has 2 copies of all of the genes. The only time that such balanced alterations lead to problems is if a chromosome happens to break right in the middle of an important gene. But the fact is that the vast majority of our DNA is just junk. That is, about 95% of our DNA is just junk and does not do anything. So most commonly, alterations or breaks in chromosomes occur in the junk DNA and don't really hurt anything directly, as long as the person continues to have 2 copies of each gene. However, in contrast to that, we will also talk about unbalanced alterations. Unbalanced alterations are ones that do result in a loss or gain of genetic material. That is, we will find individuals that might have 3 copies of some gene, or only 1 copy of certain genes. That would be an unbalanced alteration, and those patients are likely to have problems of some kind. These structural abnormalities can occur either in the germline or in somatic cells. In this study, in this book, we will mostly be talking about such alterations in the germline, those that can be passed along to future generations in the offspring. But you should be aware that these kinds of structural changes can also occur in the somatic cells. Most commonly, when they occur in our somatic cells, they are related to the development of cancer. Here are examples of an important kind of structural alteration of chromosomes. This is a type of alteration called translocations. There are 2 major kinds of translocations called reciprocal translocations and Robertsonian translocations. In reciprocal translocations, 2 different non-homologous chromosomes exchange pieces with each other. In a Robertsonian translocation, 2 different non-homologous chromosomes get stuck together. Let's look at reciprocal translocations first. This slide shows an example of a reciprocal translocation that is occurring between chromosomes number 2 and number 8. On the left part of the slide is a representation of the normal copies of chromosomes 2 and 8 that would be found in a normal individual. What is happening in this case is that one of the copies of chromosome 2 and one of the copies of chromosome 8 have broken and have exchanged pieces with each other. So the result is what we see on the right-hand part of the slide, in which we have one of those balanced alterations that we were talking about a minute ago. That is, the material in these chromosomes has been arranged in a peculiar way, a reciprocal translocation. However, this is an example of a balanced alteration in that the person in which this reciprocal translocation originally occurs is probably okay, probably normal, probably has no symptoms of any kind. And that's because this person still has 2 copies of each gene. The only time that this person would have symptoms of some kind of problem would be if one or both of those chromosome breaks occurred in the middle of an important gene. But remember, since most of our DNA is junk, most likely these breaks occur in the non-functional DNA and does not cause any problem in this person. So we would refer to a person like this as a carrier for a reciprocal translocation, because this person would not show any symptoms himself or herself, but problems could arise in the next generation when this person has children. And the idea would be the children could have problems if a child inherits an unbalanced set of genetic material. This is represented in the next slide. In the upper left part of this diagram is an illustration of what these chromosomes would look like in such a carrier for a reciprocal translocation. On the top right portion of this slide is a representation of how these chromosomes would try to pair up with each other during meiosis. In meiosis, an extremely important part of meiosis is that our homologous chromosome pairs, that is, the 2 copies of chromosome 2, the 2 copies of chromosome 8, and so forth, line up right next to each other. And in a normal individual, when the cell divides during meiosis, the chromosomes separate so that each daughter cell gets 1 normal copy of each chromosome. The problem that we have with such a reciprocal translocation, however, is that when the chromosomes try to pair with each other during meiosis, they do not form the normal, ordinary 2 pairs, and all 4 chromosomes can kind of bind together in the structure shown there in the upper right-hand part of the slide. Just to make this slide a little bit easier to talk about, this is drawn as if it were occurring in a male as he is producing his sperm. Something very similar could occur in a woman as she is producing her egg. The problem is that when that cell divides during meiosis, the chromosomes can separate from each other in several different ways, so that in the middle of the screen is shown several of the different kinds of sperm that can result from different patterns of segregation of those chromosomes during meiosis. The sperm on the left is completely normal, has 1 normal copy of chromosome 2 and a normal copy of 8, and if that sperm fertilizes a normal egg, we get the result shown at the bottom of the screen. That is, the child will be completely normal. If the second sperm has received both recombined chromosomes, but that is both translocations, and so if that second sperm fertilizes a normal egg, the resulting child will have a set of chromosomes that is just like the father. That is, that child will be a translocation carrier, will not have any symptoms himself or herself, but could give rise to problems in the next generation when that carrier grows up and has children of his or her own. The last 2 sperm shown in this diagram have received an unbalanced set of genetic material, and when either of those sperm fertilizes a normal egg, the resulting fertilized egg and the child, if it goes on to be born, will be abnormal because the resulting children will have an unbalanced set of genetic material. The third child from the left there is an example of a child who would have a partial trisomy for chromosome number 8 because the child has 3 copies of part of chromosome 8. And that child also is an example of a partial monosomy for chromosome 2 because the child has only 1 copy of a portion of chromosome 2. The child on the extreme lower right is again an example of a child with an unbalanced set of genetic material. That one would be a partial trisomy for 2 and a partial monosomy for 8. Both of those children with unbalanced sets of genetic material would have symptoms of some kind. It's impossible to predict what the symptoms would be. The children might have mental retardation, they might have some other developmental delay, but you would predict that they will have some kind of problem because they have an unbalanced set of genetic material. Here are examples of reciprocal translocation, specific examples that you should memorize for your exam. There is a reciprocal translocation that sometimes occurs between chromosome number 9 and chromosome number 22. It produces a particular characteristic chromosome called the Philadelphia chromosome. This particular reciprocal translocation leads to a disease called CML, chronic myelogenous leukemia. And that happens because this particular break does occur right in the middle of an important gene called the ABL oncogene. And by altering the structure of that oncogene, it leads to the development of this particular kind of cancer because the cell will lose control of its rate of growth. The second one on the screen is another example of a reciprocal translocation that sometimes occurs between chromosomes number 8 and 14. This one is related to Burkitt's lymphoma, and again, this particular break occurs near an important gene, in this case the MYC oncogene, and by leading to overproduction of this from this oncogene, the cells lose control of their growth, and the result is a type of cancer called Burkitt's lymphoma. The other major kind of translocation is a type called a Robertsonian translocation. In a Robertsonian translocation, two different non-homologous chromosomes get stuck together. This can only happen between two acrocentric chromosomes, as is illustrated here between chromosomes 14 and 21. In the left part of this diagram, we see the normal pairs of chromosomes 14 and 21 that we would have in a normal individual. What has happened in this particular diagram is that one of the copies of 14 and one of the copies of 21 have broken. They lose their tiny little P arms, and the two Q arms get stuck together to form the combination chromosome. The result is shown over on the right. The resulting individual has one normal copy of chromosome 14, one normal copy of 21, and the combination chromosome. So such an individual would have only 45 chromosomes. However, that person is probably okay. That is, the person in which this originally occurs probably has no symptoms of any kind, because we would call this person a Robertsonian translocation carrier. This person has no symptoms because they still have, the person still has two copies of all of the important genes from these chromosomes. For the acrocentric chromosomes, the P arms are so tiny that they do not have any important genes in them, and so it does not really hurt anything to lose those tiny little P arms. Since all the important genes for these chromosomes is on the Q arms, such a Robertsonian translocation carrier does have two copies of the Q arm of 21 and two copies of the Q arm of 14, so that person will be okay. However, where can problems develop? In the next generation, when this person has children. Again, just to make this a little bit easier to talk about, the problem here is illustrated as if it were occurring in a male as he is producing his sperm and having children, but it could equally likely occur in a female as she is producing her eggs. The upper left-hand corner of this diagram shows the chromosomes as they would exist in this translocation carrier. The part of the figure in the upper right portion of the diagram shows how these three chromosomes try to pair with each other during meiosis. Now, the problem that will occurs because we do not have ordinary normal chromosome pairs, and when that cell divides during meiosis, the chromosomes may segregate or separate improperly from each other. It turns out that those chromosomes can separate in 6 different ways to produce 6 different kinds of sperm in this individual. The part of the diagram in the upper right part of the slide shows a vertical line and a horizontal line dividing that cell into 4 quadrants. And the idea is that whatever chromosomes are found in any 2 quadrants can travel together to one of the sperm that this person will produce. We can have alternate segregation in which the chromosomes found in quadrants that are diagonally opposite each other travel together. So, for example, the first two sperm, shown there in the middle of the diagram, result from such alternate segregation. One of those sperm will have two perfectly normal chromosomes. The second one will have just the combination translocation chromosome. The other kind of segregation that can occur is called adjacent segregation. And this occurs when the whatever two chromosomes are next to each other in quadrants that are next to each other either horizontally or vertically travel together into a sperm. So you can see in this diagram that there are 4 possible kinds of sperm produced by such adjacent segregation. We will assume that this carrier mates with a normal female, and so his wife will produce a normal egg. The result in the child will depend on which of those 6 sperm wins the race to the egg. And we will get 6 different possible results. The results in the children are, the possible results in the children are shown at the bottom of this slide. On the extreme left is a completely normal child. The normal sperm combining with the normal egg will give us a completely normal diploid child. The second sperm, the one that has just the translocation chromosome, will also give us a normal child, that is, a child that will not have any symptoms. That second child will have a set of chromosomes that's exactly like the father in this case, so that child will not have any symptoms himself or herself. However, problems could occur then in the next generation when that child grows up to have children of his or her own. The third child from the left in the bottom is the one that is really important for your exam. This is a child that has a form of Down syndrome because the child has essentially trisomy 21. If you look at the chromosomes present in that child, you will find a full copy of chromosome 14 plus an extra Q arm from chromosome 14. So the child has has 2 copies of the genes on chromosome 14. The child also has 2 normal copies of chromosome 21 plus an extra copy of the Q arm from chromosome 21. So the child is effectively trisomy 21 and will have Down syndrome. Although the child has only 46 chromosomes, one of them is that combination chromosome. Approximately 5% of Down syndrome patients result from such a Robertsonian translocation. On your exam, you would be expected to know that 95% of Down syndrome patients result from the non-disjunction during meiosis we talked about earlier. 5% result from such a Robertsonian translocation. You could tell the difference between such patients by performing a karyotype. They essentially have the same symptoms, so you cannot tell just by examining the patients which type of alteration they have, but a karyotype could be used to distinguish them. It would be important to do that because of the relative risk to other members of the family. If a child has Down syndrome resulting from non-disjunction, as we saw earlier, then that child with Down syndrome was the result simply of an accident that occurred in one of the gametes that led to that child, most likely from an older mother, for example. However, that was just simply an accident in one gamete. The parents have completely normal chromosomes, and it is not likely that that same accident will happen again. So the risk to those parents of having additional Down syndrome children is very low. However, if the child has Down syndrome resulting from this Robertsonian translocation, then since one of the parents does have a chromosome alteration, it is very likely that the parents will have, if they have additional children, it is very likely that they will have an additional Down syndrome child or pregnancy losses. The last three results on the lower part of this slide would result in pregnancy losses. Monosomy 21, trisomy 14, monosomy 14. Those are all incompatible with life, and the fertilized eggs, early embryos, would be lost. There are some other common alterations in chromosomes depicted here. This type of alteration is a deletion. In a deletion, part of a chromosome is missing. We can have either interstitial deletions, in which the part that is missing from the chromosome is missing from the middle of a chromosome. We can also have a terminal deletion, in which the end of the chromosome is missing. In this particular diagram, the chromosome in the center represents a normal copy of chromosome 5. On the chromosome on the left, we have an interstitial deletion, in which the part of the chromosome that is missing is from the middle of the chromosome. And the chromosome on the right, it's the end of the chromosome that is missing. That we refer to that as a terminal deletion. Here are two examples, well-known examples of deletions that may appear on your exam. We saw earlier examples of an interstitial deletions that could lead to Prader-Willi syndrome and Angelman syndrome. These are interstitial deletions in chromosome number 15. Another example to memorize is a terminal deletion on chromosome 5 that sometimes leads to Cri-du-Chat syndrome. Cri-du-Chat syndrome is a syndrome in which the babies have a very characteristic cry that sounds like a kitten or a small cat. Another example of a chromosome alteration that we may find is an inversion. In an inversion, a portion of a chromosome is cut out, turned around backwards, and inserted upside down in the chromosome. This particular example shows a pericentric inversion on chromosome number 3. A pericentric inversion is an inversion that does include the centromere. We can also have inversions called paracentric inversions that do not include the centromere. These kinds of inversions can lead to problems during meiosis, in which pieces of these chromosomes can be lost, causing problems in the children. Another kind of chromosome alteration that can occur results in the formation of ring chromosomes. In this situation, two ends of a single chromosome get stuck together to form a circle. This is called a ring chromosome. Sometimes such ring chromosomes can be lost. So, for example, if the X chromosome were to join together to form a circle in this way and then be lost, that might be a way of creating Turner syndrome. Another chromosome alteration is a type of alteration that forms an isochromosome. An isochromosome is a single chromosome that has either two P arms or two Q arms. The chromosome on the left here depicts a normal X chromosome. What has happened in this particular case is the formation of an isochromosome containing the two Q arms stuck together. Ordinarily, in meiosis, when the two sister chromatids are separated, the complete sister chromatids separate from each other so that each daughter cell receives a complete normal chromosome. However, occasionally during meiosis, when those chromosomes are separating from each other, the separation occurs improperly, so that one daughter cell will receive the two Q arms stuck together, and the other daughter cell will receive the two X or the two Q arms stuck together. That will lead, both of those are isochromosomes. The last part of this chapter describes two relatively new advances in cytogenetics, in molecular cytogenetics, that allows us to study chromosomes in some new ways. These are advances that have been developments occurring because of the Human Genome Project. As you're probably aware, about 20 years ago, we started the Human Genome Project with the goal of determining the complete base sequence of human DNA, and we are essentially finished with that project now. That is, now we know almost the complete human DNA sequence. And as a result of what we have learned from this project, a couple of new techniques have been developed for the study of chromosomes. One of those techniques is called FISH, fluorescence in situ hybridization, and the other technique is called spectral karyotyping, or chromosome painting. Let's look at FISH, or fluorescence in situ hybridization, first. As a result of the Human Genome Project, now we know the DNA base sequence of most of our genes. And in addition, because through the technology that has been developed, we can go to the laboratory to an instrument known as an oligonucleotide synthesizer, or a gene machine, and we can get that machine to automatically make for us any single-stranded piece of DNA that we want, representing any gene that we want. And furthermore, we can get that machine to automatically attach a fluorescent label to that single-stranded piece of DNA, so that that fluorescently labeled single-stranded piece of DNA can then be used as a probe to identify the location of that corresponding gene on the naturally occurring chromosome. The idea is that a cell would be broken open to spread out the chromosomes. The DNA in those chromosomes is denatured, and the probe is exposed to those chromosomes. The probe will only be able to bind to the chromosomes at a point where the naturally occurring gene is located, and then when we shine an ultraviolet light on the chromosomes and take a picture, we will get a red spot or a yellow spot or a blue spot in the picture, that will show us the location of the gene that we were probing for. In this particular diagram, this represents an experiment in which someone was probing for a particular gene on chromosome number 14. The pair of chromosomes in the middle would represent the pair of chromosome 14 in this individual. The other two pairs are simply other pairs of chromosomes. The idea is that in this particular patient, what appeared was one single red spot, and that red spot indicates that the chromosome that it is found on is chromosome number 14, and it shows the exact location of that particular gene on chromosome number 14. The other copy of chromosome number 14 in this individual happened to have a deletion of that particular gene, and so there was no red spot appearing on that copy of chromosome number 14. In a normal individual, we would have seen two red spots, one on each copy of chromosome 14. So such FISH probes, fluorescence in situ hybridization, can be used to identify small deletions, for example, in patients. They can also be used to easily identify trisomies, for example. A patient with Down syndrome would show 3 fluorescently labeled spots if we probed for a probe on chromosome number 21. The other important technique that has been developed is called spectral karyotyping, or chromosome painting. In this technique, that again grew out of the Human Genome Project. A series of probes have been developed such that when a mixture of chromosomes is exposed to this mixture of probes, and then an ultraviolet light is shown on the chromosomes and a picture is taken and the results are analyzed by a computer, each individual chromosome is painted its own separate specific color, and it becomes very easy to distinguish each chromosome from the others. It's a much easier technique for identifying specific chromosomes compared with the banding patterns that we saw at the beginning of this chapter. In this particular diagram, this represents an individual in which a reciprocal translocation has occurred. And you can see that there has been a reciprocal translocation between one of the red chromosomes and one of the dark blue chromosomes. These color differences are extremely easy to see. And it's much easier to identify the problems in our patients compared with having to analyze the banding patterns as we saw earlier in the chapter. Okay. In this chapter on cytogenetics, we've talked about numerical abnormalities of chromosomes. We've talked about structural abnormalities of chromosomes. You're very likely to see examples of these on your exam. Be sure that you memorize the common examples that we've talked about. Be sure that you can study a karyotype and identify particular syndromes associated with the alterations that we've talked about. We're continuing our discussion of medical genetics. I'm Dr. Reichenbaker from Marshall University, and in this segment we'll be talking about Chapter 4 in your book, The Genetics of Common Diseases. By common diseases, we mean diseases such as heart disease, cancer, and Alzheimer's disease, diseases that are very common in the population. The genetics of these kinds of diseases is very complex because there are typically many different genes and many different environmental factors involved. The kind of inheritance that these diseases exhibit is something called multifactorial inheritance. By multifactorial inheritance, we mean that these diseases, like heart disease and cancer, for example, are caused by many different factors. Typically, there is a polygenic component and an environmental component. That means there are typically many different genes involved and many different environmental factors involved as well. These kinds of traits and diseases follow a bell-shaped curve or normal distribution when they are illustrated in a graph form. Some examples of these kinds of diseases some examples of these kinds of multifactorial traits are things such as diastolic blood pressure, height, weight, IQ, and body mass index. And an example of how we can display one of these multifactorial traits in graphic form is shown in this diagram. At the top of the screen, we're showing a graph of body mass index, or BMI, and we're graphing on the y-axis the number of individuals in the population with each of the exhibited BMIs. And what this diagram illustrates is that when we graph one of these multifactorial traits, there is a normal distribution, bell-shaped curve type of distribution in the population. That is, in the large population, there will be many possible BMIs, and they will be distributed widely, in a widely varying way, over the population. The graph at the bottom of the screen illustrates how these kinds of traits, such as body mass index, can be related to a disease such as obesity. For a typical common disease such as obesity, we will define that disease as a body mass index of 30 or above, so that there is a particular threshold of body mass index that is used to indicate the condition of obesity. So that in that graph at the bottom of the screen, everyone who has a body mass index above a level of 30 will be considered to be obese. So everyone in the black part of that curve is said to be obese. Now, the way that this kind of concept is related to the common diseases such as heart disease, cancer, and so forth is illustrated in this next diagram. This diagram illustrates a concept called the multifactorial threshold model for diseases such as heart disease. On the y-axis in these diagrams, we are still graphing numbers of individuals. On the x-axis, we are graphing liability for the development of one of these diseases, such as heart disease. Liability could also be considered risk or susceptibility. What liability really means is how many bad genes and how many bad environmental factors do you have towards the development of a disease such as heart disease. The idea is that someone who is very low on the liability scale would have lots of good genes and lots of good environmental factors. Someone who is very high on the liability scale would have lots of bad genes and bad environmental factors towards the development of heart disease, for example. The idea here is that there will be some threshold of liability that is some combination of bad genes and bad environmental factors, such that everyone in the population above that particular threshold will have a disease like heart disease. And so in these graphs, everyone above that threshold of liability, everyone in the black part of this curve will have heart disease. And this diagram also illustrates that threshold of liability for a disease like heart disease can be different in between men and women. At the top part of the screen is shown the threshold of liability for men. The graph at the bottom shows the threshold for women. Women have a higher threshold of liability for heart disease. So, for example, what that basically means is that it's more difficult for a woman to develop heart disease than it is a man, so that a woman must have more bad genes and or more bad environmental factors before she will develop heart disease. Now, one of the kinds of things that we want to be able to do with these multifactorial diseases, such as heart disease, is to predict the recurrence risk. That is, at some point, someone will come into your office and will sit down at your desk and will say, "Doc, what are my chances of developing heart disease?" How are you going to answer that question? It turns out that answering that question is a lot more difficult than it is for predicting the recurrence risk for those simple single gene diseases that we talked about in earlier segments of this course. With the simple single gene diseases such as cystic fibrosis and so forth, we can, by knowing the genetic status of the parents, we can make very definite mathematical predictions about the recurrence risk in those kinds of families. However, it's much more complicated for a disease such as heart disease, in which there are many different genes and many environmental factors involved. So let's talk about some of the things that you have to take into consideration in order to make recurrence risk predictions for an individual. The recurrence risks for multifactorial diseases such as heart disease are estimated empirically. That means by observation. That means you cannot do a simple mathematical calculation. It basically means you have to do epidemiological type studies. You have to go out into the population and collect a lot of data. And essentially what people do is to go out into the population and find someone who has heart disease and ask them a lot of questions. Do you have any brothers with heart disease? Do you have any sisters with heart disease? What about your father? What about your mother? And then by collecting all that large amount of information from a large population, then when that one person comes down and sits at your desk, you try to relate that one person's family situation to all that data that has been collected from a large population. That's what we mean by estimating the risk empirically. The recurrence risk increases as the number of affected relatives increases. That means I'm at a higher rate for developing heart disease if I already have a brother who has heart disease. And I'm at an even higher risk if I have two brothers with heart disease. The idea there is, if I already have several brothers with heart disease, my brothers, in order to have heart disease, have to be far to the right on that liability scale. They have lots of bad genes and lots of bad environmental factors. And if I have several brothers with bad genes and bad environment, I probably do too. That places me at higher risk. The risk increases as the severity of the disease expression increases. That means I'm at a higher risk for developing heart disease if I have a brother with very severe heart disease compared with having a brother who has only mild heart disease. That's basically because the severity of the symptoms usually are related to how far to the right on that liability scale a person is. So if I have a brother with very severe heart disease, he must have lots of bad genes, lots of bad environmental factors, and if my brother has that, I probably do too. It puts me at higher risk. The risk increases if the proband is a member of the lower risk gender. The proband is the first person in a family that you find with some disease. For heart disease, the lower risk gender would be females. That is, if you remember from the earlier diagram, the threshold of liability is higher for females than it is males for heart disease. And so what this item basically means is I am at a higher risk for developing heart disease if I have a sister with heart disease compared with having a brother with heart disease. The idea there is, if I've got a sister with heart disease, in order for her to have the disease, she has to be very far to the right on the liability scale, lots of bad genes, lots of bad environmental factors. If my sister has that, I probably do too, puts me at high risk for developing heart disease. The risk decreases very rapidly for more remotely related relatives. That means that for diseases such as heart disease, really the close relatives are the only ones that we really count when we're estimating risk. Typically, siblings, brothers and sisters, and parents. It really doesn't matter what your third cousin has. It's your closely related relatives that are those that are of use in making predictions about your risk. And finally, the risk increases as the prevalence of the disease increases in a population. That one basically means that I am at a higher risk for developing heart disease just simply because I live in the United States compared with living in Japan, for example. For one reason or another, heart disease is less common in Japan than it is in this country. So simply because I live in the United States, I am at a higher risk for developing heart disease. Now on the exam, if they ask a question about these general principles, they have to keep it fairly easy. That is, you have to easily be able to distinguish between the options. For example, I can imagine a test question in which they might ask something like, which of the following people is at highest risk for developing heart disease? A person who has one brother with heart disease. B, a person who has two brothers with heart disease. Or C, a person who has two sisters with heart disease. In that example, obviously the answer would be C, the person who has two sisters with heart disease. They cannot ask you to distinguish between two sisters versus four brothers or something like that. They have to keep it fairly simple. So if you know these basic principles on this screen, you should be able to answer the types of questions that they would ask about this topic. How have people figured out the relative influence of genes versus environment on common diseases like heart disease and cancer? That is, how have they determined nature versus nurture? There have been two major kinds of studies that have been used: twin studies and adoption studies. Let's look at twin studies first. In twin studies, what is compared is the situation in monozygotic or identical twins who are genetically identical, they share all of their genes, compared with dizygotic or fraternal twins who share only half of their genes. Dizygotic fraternal twins are no closer genetically than any other brother and sister. They share only half their genes. What they look for in twin studies is concordance. That is, they will find a twin who has some disease, like heart disease or breast cancer or any of the common diseases. They will find a twin who has one of those diseases, and then they will ask, does the other twin also have the same disease? That is, they are measuring concordance. The idea here is that if a trait or a disease is completely determined by genes, then for monozygotic twins, concordance would be 1 or 100%, since they share all their genes. However, for fraternal twins, for such a disease, concordance would only be 50%, 0.5 or 50%, since such fraternal twins share only half their genes. So these kinds of tests have been done for a large number of traits and diseases, as shown on this slide. This slide shows on the left a large number of traits and diseases that have been examined. The concordance rates have been determined in monozygotic twins, the column in the middle, and dizygotic twins, the column on the right. If you look down through that long list, what you will find is that the concordance rates for all these items, the concordance rates are higher in the monozygotic, identical twins, than they are in dizygotic, fraternal twins. However, we do not find the numbers 1 and 0.5. So what this indicates is that for all the diseases and traits in this list, genes play a role, but they're not the whole story. Environmental factors are also important. We can get an idea of the relative importance of genes as opposed to environmental factors by looking at the differences in concordance rates in the monozygotic and dizygotic twins. For example, the trait of height, about halfway down the list, shows a concordance rate in monozygotic twins of 0.94, but only 0.44 in dizygotic twins. There's a relatively large difference in those concordance rates. A large difference in concordance rates indicates that genetics plays a relatively large role. So that would indicate that height is influenced more by genes than it is by environment. On the other hand, measles, for measles, you will find that the concordance rates are not very different in monozygotic and dizygotic twins. There's only a slight difference in concordance rates. That indicates that for a disease such as measles, the environmental factors play a much greater role. Adoption studies have also been applied to determining the influence of genes as opposed to environment. When adoption studies are done, again, what is measured is concordance, and what they do is compare the traits and diseases in adopted individuals with those traits and diseases of their adoptive families and their biological families. The results are very similar to what we saw with the twin studies. That is, for a large number of traits and diseases, genes are involved, but environmental factors are also involved. Many of these common diseases have both a familial or inherited form and a sporadic form. That is, most cases for the common diseases such as heart disease, cancer, Alzheimer's disease, and so forth are sporadic. That is, there is not a clear inheritance pattern in particular families. However, for many of these diseases, there will also be special familial or inherited cases, which are much more rare. That is, for many of these diseases, we can find particular families in which there is a very strong inherited mutation that leads to the disease. Sometimes the particular genes that are mutated in the familial cases are also the same ones that are important in the sporadic cases of the diseases. For example, mutations in a gene called P53 would be such an example. P53 is a gene, is a tumor suppressor gene related to the development of cancer. It turns out that there are certain families in which a mutated form of P53 is inherited, leading to a strong relationship to cancer in those families. But it also turns out that mutations in P53 are important in many of the sporadic cases of cancer as well. That is, we have found mutations in P53 in approximately half of all cancers. Sometimes those genes mutated in the familial cases, however, appear not to be so important in the sporadic cases. An example of that would be the breast cancer-related genes BRCA1 and BRCA2. The inherited forms of breast cancer are caused by mutations in those two genes, but it turns out that sporadic cases of breast cancer only rarely have mutations in those same genes. The sporadic cases of breast cancer are caused by mutations in many other genes. Let's look at some specific examples of multifactorial diseases and particular genes associated with them. Let's look first at some inherited cancer syndromes. Inherited cancer syndromes usually result from germline mutations, that is, those that can be passed along from generation to generation, in either tumor suppressor genes or proto-oncogenes. For your exam, you will be expected to know the basic features of proto-oncogenes and oncogenes and tumor suppressor genes. The basic features of those kinds of genes are shown in this table on your screen. The idea here is that we all have two perfectly normal kinds of genes that are involved in the normal regulation of normal growth of our cells. These two kinds of perfectly normal genes are called proto-oncogenes and tumor suppressor genes. Proto-oncogenes are genes that produce a protein that one way or another is involved in stimulating the growth of cells, making cells grow faster. Tumor suppressor genes code for proteins that are basically involved with slowing down or inhibiting cell growth. And the idea is that in our normal cells, we have a balance between the proto-oncogenes speeding the cell up, tumor suppressor genes slowing growth down, so that in our normal cells we have a good balance between those two genes and our normal cells grow at the correct rate of speed. The analogy I've heard to this that I like involves driving your car down the road. How do you maintain the speed of your car at the correct speed? Well, you need to use both the accelerator, the gas pedal, and the brakes. You need to use them both, and you need to use both of them in a balanced fashion in order to keep your car moving the correct rate of speed. How can you lose control of the speed of your car? Well, one of the ways that you might lose control of your car is if the gas pedal, the accelerator, got stuck to the floor. Having the gas pedal stuck to the floor is sort of like having a mutation in a proto-oncogene that converts it to a cancer-causing oncogene. Such a mutation will lead to a turn-on of cell growth. That is, that mutation will cause the cell to grow faster. It will promote tumor growth. These kinds of mutations, mutating a proto-oncogene to an oncogene, is typically a gain-of-function mutation. That is, we have either created a new enzyme or are producing too much of a growth stimulatory protein, leading to a speed-up of cell growth. Another general principle of such oncogene mutations is that this is a one-hit phenomenon. That is, remember, we have 2 copies of almost all of our genes, but for oncogene mutations, we only have to mutate 1 copy of a proto-oncogene, converting it to an oncogene, in order to lead to the development of cancer. What's the other way we can lose control of the speed of our car? Brake failure. Brake failure is sort of like mutating or deleting our tumor suppressor genes. If we delete or mutate tumor suppressor genes, then we can no longer slow down cell growth. The cells will grow out of control and lead to the development of cancer. Mutations in tumor suppressor genes are typically loss of function mutations. That is, an enzyme or a protein is missing from the cell. And another general principle of mutations of tumor suppressor genes is that this is a 2-hit phenomenon. That is, we have to mutate or knock out both copies of the same tumor suppressor gene within a single cell in order to lead to the development of cancer. So be sure you know all these basic principles involved in oncogenes and tumor suppressor genes. You will be expected to know them for your exam. Let's look at some particular examples of types of inherited cancer and see how they're related to those basic principles. In an earlier section, we talked about an inherited form of cancer called retinoblastoma, which is cancer of the developing retina. And earlier on, we said that such inherited retinoblastoma is caused by mutations of the RB tumor suppressor gene. This disease is inherited in an autosomal dominant fashion with 90% penetrance. And you might remember that in that earlier section, we said that for the inherited form of retinoblastoma, the first hit was inherited, and the second hit or mutation is acquired. That is, early in the development of the retina, if a person who is heterozygous for such an RB tumor suppressor mutation, if early in the development of that retina, one single cell acquires a secondary somatic cell mutation that mutates or knocks out the other copy of the RB gene within that one cell, one single cell, then that one cell has lost its heterozygosity and has 2 mutations in both copies of the RB tumor suppressor gene, and that one single cell then will multiply, grow out of control, and develop into a tumor. Here are some other examples of tumor suppressor genes that are mutated in inherited forms of cancer. BRCA1 and BRCA2 are tumor suppressor genes related to breast cancer. In families in which breast cancer is being inherited, what a woman will inherit is 1 mutant copy of BRCA1 or 1 mutant copy of BRCA2. If any time during her lifetime, then, she acquires a secondary somatic cell mutation in a breast cell that knocks out the other copy of the same mutated tumor suppressor gene, then that one single cell will lose control of its growth and will develop into the breast tumor. If the woman originally inherited a mutation in a BRCA1 gene, then her secondary acquired mutation has to knock out the other copy of BRCA1. If, however, her first inherited mutation was in BRCA2, then the second mutation has to knock out the other copy of BRCA2. That is, both mutations have to be in the same tumor suppressor gene to lead to the development of cancer. Another important, very important tumor suppressor gene is P53. We've found mutations in P53 in about half of all cancers. There is an inherited cancer syndrome related to mutations in P53. It's called Li-Fraumeni syndrome. The pattern of inheritance here is just like it is for retinoblastoma and inherited breast cancer. That is, what these people in these families inherit is one mutant copy of P53. They are heterozygous in all the cells of their body. However, if any time during their lifetime they acquire a secondary somatic mutation that knocks out the other copy of P53 within any cell of the body, that one cell will lose control of its growth and will develop into a tumor. If that secondary mutation occurs in the lung, then the patient will have lung cancer. If the secondary mutation occurs in the liver, the patient will have liver cancer, and so forth. Li-Fraumeni patients typically have many, many tumors scattered all over their bodies, just depending on where those secondary mutations occur. This slide also shows you a couple of other important tumor suppressor genes and their relationship to particular types of cancer. There's an inherited form of melanoma that involves mutations in a tumor suppressor gene called P16. There's also an inherited form of colon cancer that involves mutations in a gene called APC. In all of these cases with inherited forms of cancer involving tumor suppressor genes, in all the cases, the person inherits one mutant copy of the gene, and then they will develop cancer if they acquire a secondary mutation that knocks out the other copy of that gene, of that same tumor suppressor gene within one single cell. Here are some examples of particular genes that we have found associated with some other common multifactorial diseases. Coronary heart disease, for example. There is an inherited form of coronary heart disease that is related to mutations in the LDL receptor gene. As you might imagine, if we have mutations in our LDL receptors, we cannot metabolize LDL properly. That will lead to an elevated level of cholesterol in the bloodstream, and that elevated level of cholesterol will lead to an increased rate of heart disease and an increased risk for heart attacks. Alzheimer's disease also is one of these multifactorial diseases, and by studying families in which Alzheimer's disease is inherited, we have identified 3 genes: BAP, presenilin, apolipoprotein E, that are related to the development of Alzheimer's disease. Our study of these multifactorial diseases is still in its early stages. That is, these common multifactorial diseases have many, many genes and many, many environmental factors related to them, and we are just barely beginning to learn the identity of these genes and environmental factors. Here are some of the basic principles that we've seen related to these common diseases. These common multifactorial diseases have both a genetic and an environmental component. Their distribution in the population follows a bell-shaped curve or normal distribution. For each of these multifactorial traits or diseases, there is a particular threshold of liability. That is, for heart disease, for example, there is some combination of bad genes and bad environment that is required in order for a person to develop heart disease. We've also seen that the recurrence risks are determined empirically. That is, we must do population studies to make predictions of recurrence risk for these diseases. There are no simple calculations that we can do. And we've also seen that for these common diseases, there are familial, strongly inherited versions of these diseases, but the vast majority of these diseases occur sporadically. In some cases, the genes involved with the familial cases are the same as the genes involved with the sporadic cases, but in other cases, those genes are different as well. We're continuing our discussion of medical genetics. I'm Dr. Vernon Reichenbaker from Marshall University. We're continuing our discussion by covering the topic of gene mapping in Chapter 5 in your book. An important aspect of understanding genetic diseases is knowing what genes are involved with those diseases, identifying their locations, and cloning of those genes. In this chapter, we'll learn about how we can identify the location of genes and use that information for the cloning of such genes. Gene mapping is also known as linkage analysis. With the techniques of gene mapping or linkage analysis, we determine basically two things. We can determine where a gene is located. That is a particular gene found on chromosome number 6? Exactly where on chromosome number 6 is that gene found? Another aspect of gene mapping or linkage analysis is identifying the distance between two different genes. That is, are two genes close together or are two genes far apart? The importance of this information is that knowing the location of genes can help us to clone genes so that we can study and identify those genes, identify mutations in genes related to genetic diseases so that we can learn more about how mutations cause diseases and maybe how we can fix the problems. And another important aspect of gene mapping, or a product of gene mapping, is genetic diagnostic tests. That is, the development of DNA testing or gene testing or genetic testing for the identification of people who are carriers for mutations causing genetic diseases. We'll talk in this chapter about the basic techniques for mapping genes, determining their location. We'll also talk a little bit about how that information is useful in the cloning of genes. We will talk about the genetic diagnostic tests in Chapter 6. What kinds of genes are most commonly mapped when we do gene mapping or linkage analysis? Theoretically, any kinds of genes can be mapped. However, most of the time when we do gene mapping or linkage analysis studies, we most commonly use a type of gene called a DNA marker. This diagram on your screen illustrates for you 4 different kinds of DNA markers that are commonly used in gene mapping experiments. DNA markers are particular kinds of genes or segments of DNA in our genomes that are very highly polymorphic. These DNA markers don't necessarily do anything. That is, they do not necessarily code for proteins. They do not necessarily have any function at all. But their importance is that we can identify them and test for them in the laboratory, and these DNA markers are very highly polymorphic in the population. That is, there are many variations of these genes in the population that make them useful for gene mapping. In panel A in this diagram is illustrated a type of DNA marker called an RFLP, a restriction fragment length polymorphism. This diagram basically indicates that some of us in the population might have a DNA molecule that can be cut, for example, in 3 places by a restriction endonuclease such as EcoR1 to give us a particular pattern of DNA products. different sizes of DNA restriction fragments, whereas others of us in the population might have a different DNA sequence in that region, such that that DNA molecule is cut differently by the restriction enzyme, giving a different pattern of sizes of fragments of DNA. So the difference in the population between these two pieces of DNA is the difference in sizes of the DNA molecules that are produced by cutting with the restriction enzyme. We can detect those differences in the lab using a technique called Southern blotting. A second kind of DNA marker often used in gene mapping studies is shown in panel B. This is a type of DNA marker known as a VNTR. That stands for Variable Number of Tandem Repeats. This kind of DNA marker is also sometimes known as a mini satellite. The idea here is that there are many places in our genomes where we have a repeated DNA sequence. That is, a base sequence of DNA that is repeated several times in a row. That would be known as a tandem repeat. In this diagram in panel B, each one of those little rectangles represents such a repeated sequence. Each one of those little boxes might represent a base sequence of about 50 or so base pairs. And the idea is that some of us in the population might have a DNA molecule that contains 6 of those repeated segments in a row, whereas others of us in the population might have a DNA molecule with only 4 of those repeated sequences in a row. Other people in the population might have 10. This site could be highly polymorphic. There could be many variations in the population. This is perfectly normal. We all have lots of these in our genomes. The difference in these DNA sequences or DNA markers is the size of the piece of DNA involved. And again, we can measure these differences in size in the laboratory by the technique called Southern blotting. Panel C shows another kind of DNA marker often used in mapping. This is a kind of marker called an STR or STRP, which stands for short tandem repeat polymorphism. This kind of marker is also sometimes known as a microsatellite. STRs are just like VNTRs, except they're smaller. In STRs, each of the repeated sequences might be 2 or 3 or maybe up to 6 base pairs long, much smaller repeated segments. And in the population, some of us might have a segment of DNA that has this sequence repeated 5 times. Other people might have this sequence repeated only 3 times, others 10 times, others 15 times. Again, we have a polymorphism, a highly variable polymorphism in the population. The difference between the different DNA molecules in different people will be the difference in the size of this DNA marker. STRs are smaller than VNTRs, so we typically will detect these STRs using the polymerase chain reaction, or PCR. The last example in panel D is known as an SNP, a single nucleotide polymorphism. This just simply means a single base change. That is, some of us in the population might have a G at some particular location, others might have a C. These SNPs can be detectable either by DNA sequencing or by ASO probes, allele-specific oligonucleotide probes. Now, as a result of the Human Genome Project, we have identified something like 3 million of these DNA markers in our genomes. And as a result of that Human Genome Project, now we know where all these markers are and we know what they are. And with our laboratory tests, we can identify them in the lab. In genetic studies, we can use these DNA markers just like mile markers on an interstate highway. If you're driving down an interstate highway and look over to the right-hand side of the road, you will perhaps see a little green sign that says mile 121. Then you go a mile, it says mile 122, and so forth. Those mile markers tell you where you are on that highway. We can use these DNA markers, just like those mile markers on the highway, to tell where we are on a chromosome. As we're studying particular DNA molecules or chromosomes, by detecting these markers, we can know where we are on that chromosome. There are basically two major approaches to using these kinds of DNA markers and other genes for the mapping of genes. These two major approaches involve determining recombination frequency, and also the other approach is to measure the amount of linkage disequilibrium. Let's look at the recombination frequency approach first. The idea behind using recombination frequency is this. If there are two particular alleles in a parent, we can determine whether those two alleles are both transmitted together to offspring, or whether those two alleles become separated when they are transmitted to an offspring. And the frequency with which those two alleles become separated from each other when transmitted to an offspring is called the recombination frequency, and this recombination frequency can give us a measure of the distance between two different genes. If the two genes that we're looking at are on two completely different chromosomes, then we say that those two genes are unlinked and they show 50% recombination. That is, two genes that are on two completely different chromosomes are inherited independently of each other. They are inherited randomly with respect to each other, and those two genes will only travel together from parent to offspring half the time. So we say that in that situation we have 50% recombination. If two genes are on the same chromosome but far apart on that same chromosome, we will still get 50% recombination when those genes are transmitted from the parent to a child. And we will say that those genes are unlinked if they are far apart on the same chromosome. The idea is that if two genes are far apart on the same chromosome, that there will be enough crossing over between those two genes to allow them to be randomly inherited from parents to the children, such that, again, we will get 50% recombination and the genes will be said to be unlinked. However, if two genes are located close together on the same chromosome, then we say that those two genes are linked, and we will find less than 50% recombination when those genes are transmitted from the parent to the offspring. The idea here is that if two genes are close together, crossing over during meiosis will tend to occur outside the region of those two genes, and they will tend to both stay together as they are transmitted to the offspring. We will get a recombination value of perhaps 20% or 25%, something less than 50%, indicating genetic linkage and the fact that those two genes are close together on the same chromosome. These principles are illustrated in the next series of slides. This slide illustrates two genes that are on two completely different chromosomes. The two genes depicted here are known as gene G and gene M. And for each of those two genes, there are two possible alleles. That is, in the individual depicted here, the person on one particular chromosome has a copy of allele G1 on one of the chromosomes and allele G2 on the other chromosome. On a completely different pair of chromosomes, is found gene M, and this individual has two alleles for gene M, known as M1 and M2. If we want to determine the frequency with which alleles G1 and M1 are transmitted to the child from the parent, we will find that those two alleles will travel together to the child only 50% of the time. That is, genes G and gene M, because they are located on two completely different chromosomes, these genes will be inherited randomly with respect to each other, and allele G1 will only stay together with allele M1 half the time. They will be separated half the time. We call that 50% recombination, and we say that those genes are unlinked. In this diagram, panel B, we're illustrating two possible genes that are both located on the same chromosome, but far apart on that chromosome. And the idea is that this individual contains alleles G1 and M1 on one copy of that chromosome, and the individual has alleles G2 and M2 on the other copy of that chromosome. Because these two genes, G and M, are far apart on this chromosome, when those two chromosomes line up next to each other during meiosis, crossing over will occur, and because that crossing over means that the chromosomes will literally cross over and exchange pieces with each other, and that this can occur between all of our homologous chromosome pairs and typically occurs many times along the length of a chromosome. In this example, since genes G and M are far apart on the same chromosome, there may be several crossovers that occur during meiosis as this person is producing his or her gametes. And because there are many opportunities for crossing over between those two genes, gene G and M will be inherited randomly with respect to each other again. That is, if we follow the inheritance of allele G1 and M1, they will stay together when transmitted from the parent to the offspring only half the time, but the other half the time they will become separated during inheritance. So again, since they are inherited randomly with respect to each other, there will be 50% recombination, and we say that those genes are unlinked. Panel C shows an example in which the two genes G and M are located close to each other on the same chromosome. In this case, this individual has alleles G1 and M1 on one copy of this chromosome pair, and alleles G2 and M2 on the other copy of this chromosome pair. The idea here is that because genes G and M are close together on the same chromosome, that when crossing over occurs between these two chromosomes, the crossing over is most likely to occur outside the region of those genes, and it is not very likely that a crossover will occur between them. And because the crossovers tend not to occur between them, alleles G1 and M1, for example, will tend to stay together most of the time, and they will be separated or will undergo recombination less than 50% of the time. So in this kind of example, we say that recombination is less than 50%, and we say that these two genes are linked. So the bottom line here is that if two genes are linked, that means we get recombination less than 50% of the time, and those two genes are close together on the same chromosome. We can use the frequency of recombination to get an estimate of the distance between two genes. If we find a recombination of 0, that means that the two genes are very, very close together. However, if we find a recombination frequency of 50%, it means the two genes are very far apart. Any recombination value between 0 and 50% will give us a measure of the distance between those two genes. The smaller the recombination frequency, the closer the two genes are to each other. Here's an example of how we might determine the recombination frequency between two genes. In this particular pedigree, we are going to be following the inheritance of two different genes. One of these genes is the neurofibromatosis type 1 gene, and there will be two possible alleles for this neurofibromatosis gene. There will be a normal allele and a mutant allele for the neurofibromatosis gene. Since neurofibromatosis type 1 is an autosomal dominant disease, we can determine whether a person has inherited the normal or the mutant alleles by whether or not they are affected by the disease. The second gene that we will follow the inheritance of in this pedigree is an RFLP gene, one of those restriction fragment length polymorphisms. And for this particular RFLP gene, there are two different possible alleles in this family. And we simply call those two alleles allele 1 and allele 2. We can follow the inheritance of the RFLP alleles by detecting the different sizes of bands on a southern blot when we look at the DNA from each member of the family. In this pedigree, the numbers underneath each symbol shows the genotype for the RFLP gene that we will follow the inheritance of. What we are going to do in this family is follow the inheritance of these two genes to determine the recombination frequency to get a measure of how close together or how far apart the neurofibromatosis gene and the RFLP gene are to each other. What we're going to follow in this family is just the inheritance of these genes from the mother in the second generation to her 6 children in the third generation in this pedigree. We don't care about anything else. All we care about is inheritance of these genes from this mother, second generation, to her 6 children in the third generation. The first thing that we have to figure out is the linkage phase for these two genes in this woman in the second generation. That is, we have to figure out her haplotypes, that is, how those particular alleles for these genes are arranged on her copies of chromosome number 17. Chromosome number 17 is the one that carries the neurofibromatosis type 1 mutation. By looking at this pedigree, you should be able to see that from her father, the woman in generation 2, from her father, she must have inherited a normal neurofibromatosis allele, and she must have inherited allele 2 of the RFLP. From her mother, she inherited the mutant neurofibromatosis allele, and she must have inherited allele 1 of the RFLP marker from her mother. So that we would say that the haplotypes for chromosome 17 for this woman are indicated in this diagram. By the pattern of inheritance from her parents, we can see that one copy of chromosome 17 in this woman carries the mutation for neurofibromatosis and allele 1 of the RFLP gene. Her other copy of chromosome 17 carries the normal allele for neurofibromatosis and RFLP allele number 2. Remember The neurofibromatosis gene and the RFLP gene are two completely different genes. Now what we're going to do in this family is follow inheritance of these alleles for these two different genes from this mother to her 6 children. And we will be asking the question, do those alleles stay together as they are arranged on the mother's chromosomes when they are passed to each child, or do they undergo recombination or rearrangement or crossing over when they are passed from this woman to her 6 children? Notice that each of the 6 children in the 3rd generation must receive a normal neurofibromatosis allele and allele 1 of the RFLP marker from their father. All 6 children get the same thing from their father, so all we care about is what comes from this mother. The idea here will be, if a child inherits the mutation and RFLP allele number 1, then there was no crossing over, no recombination during meiosis in the egg that led to that child, and we say there was no recombination in that child. If a child inherits the normal neurofibromatosis allele and RFLP allele number 2 from the mother, then again we say that there was no crossing over or recombination between those two genes during meiosis in the egg that led to that child, because the child would have received the same arrangement of alleles from the mother as they originally occurred in the mother. However, if a child inherits a copy of the mutation from the mother along with RFLP allele number 2, or if a child inherits from the mother the normal neurofibromatosis allele along with RFLP allele number 1, then recombination or crossing over must have occurred between those two genes on those chromosomes. That is, during meiosis, when the woman was preparing the egg that led to that child, there would have been an exchange of material between those chromosomes so that the child would receive a different combination of alleles compared with the way they were arranged in the mother. Now let's look at those children and see whether or not we find any recombination. If we look at the 3 affected children in generation 3, we can see that those 3 affected children must have received a copy of the mutation from their mother. And since the genotype for the RFLP in each of those children is 1-1, they must also have inherited allele number 1 of the RFLP from their mother. That is, each of those children inherited the mutation and allele number 1 from their mother. That was the same arrangement in which those alleles occurred on the mother's chromosome, so there was no recombination or crossing over between those two genes in the eggs that led to any of those 3 affected children. If we look at the 2 normal daughters in generation 3, we can see that they must have inherited a normal neurofibromatosis allele from their mother, and we can also see that they must have inherited allele number 2 of the RFLP marker from their mother. Since they inherited the normal allele and RFLP allele number 2, they have received an arrangement of alleles that's exactly the same as the arrangement in the mother, so there was no recombination, no crossing over in the eggs that led to those 2 normal daughters. Now let's look at the normal son in the far right of generation 3. This normal son must have inherited the normal neurofibromatosis allele, since he does not have the disease, but because his genotype for the RFLP marker is 1-1, he must have inherited from his mother allele number 1 of the RFLP marker. So he inherited the normal allele and allele number 1 of the RFLP marker from his mother, but in his mother, those alleles were on 2 different chromosomes. The only way that normal son could have inherited that particular combination of alleles would be if there was a crossover event or an exchange of material between those two chromosomes during meiosis as the mother was producing the egg that led to that normal son. Because we did find such recombination in that son, we refer to him as a recombinant. And notice that we found between those two particular genes that we're looking at in this family, we found recombination occurring in one child out of 6. Since we found recombination in one child out of 6, we say that the recombination frequency between those two genes in this family was 1 divided by 6, or 17%. That 17% recombination frequency gives us a measure of the genetic distance between those two genes, the neurofibromatosis gene and the RFLP gene. Remember, if we had found a recombination frequency of 0%, we would say that the two genes are very close together. If we had found a recombination frequency of 50%, we would say that the two genes are far apart. Since we actually found 17% recombination, that tells us that the genes are linked and they are sort of close together, but not really, really close together. Genetically speaking, we refer to each percent recombination as a centimorgan. So we would say that these two genes are 17 centimorgans apart, and we now know that a rough estimate is that each centimorgan is worth about 1 million base pairs. So taken at face value, that would say, this information would tell us that these two genes are approximately 17 million base pairs apart. Now the problem with this number, as we've determined here, is that we have only been looking at one small family, one family with 6 children. That is too small a number to get accurate information. We can get more accurate information, more accurate estimates of genetic distances by simply looking at larger numbers. Now, we're not going to make this one poor woman have 100 kids, but what we're going to do is go out into the population and find many families in which these two genes are segregating, determine the recombination frequencies from all those large number of people in the population. We will collect all that data and feed that data into a computer. The computer will then go through a complicated calculation and will calculate something called a LOD score. LOD stands for logarithm of the odds. And such a LOD score basically gives the computer's best estimate, best determination of whether or not two genes are actually linked, and it also will give the computer's best estimate of the real, actual recombination frequency between two genes. The key to understanding LOD scores is shown at the bottom of the screen. No one will ever ask you to calculate a LOD score because the calculation is just too complicated. Computers do the calculation, but you will be expected to know the interpretation of a LOD score. If the computer tells you that the LOD score is greater than 3, that your conclusion is the two genes are linked. If the computer tells you that the LOD score is less than -2, your conclusion is the two genes are not linked. If the computer gives you a value between 3 and -2, it means that you don't have enough data yet, that you have to collect more information and keep giving information to the computer until the computer determines a LOD score above 3 or below -2. How might you see this kind of information on the USMLE? On the USMLE, you might see LOD scores presented in, as is indicated on this slide. That is, what the computer has done mathematically is to mathematically test a whole series of different theta values or recombination frequencies. and it has determined a LOD score for each possible recombination frequency. The only thing that you look at in such a table is the highest LOD score. Forget about everything else in the table, but just look at the highest LOD score and the particular theta value or recombination frequency that gave that highest LOD score. The highest LOD score is the one that will be most accurate, and so our determination is made from that one. In this particular example, we do find a LOD score of 3.5. That is, we do find a LOD score greater than 3, and so our conclusion here would be, yes, these two genes are linked. The actual recombination frequency, that is the computer's best estimate of the recombination frequency, is the theta value that gives us that highest LOD score. In this particular case, the LOD score of 3.5 was obtained using a recombination frequency of 0.1, so we would determine that the actual recombination frequency between these two genes is 0.1 or 10%. So our conclusion is, yes, these two genes are linked. Their recombination frequency is 10%. These two genes are 10 centimorgans apart, and we would conclude that these two genes are located about 10 million base pairs apart. A second way, or a second approach to determining linkage between two genes is the concept of linkage disequilibrium. Linkage disequilibrium occurs if the particular alleles for two linked genes are seen together more frequently or less frequently than would be predicted by chance alone. The bottom line conclusion is the greater the linkage disequilibrium you find, the more closely linked the two genes are. An example of this is shown in the next diagram. The part of the picture near the bottom of the screen is a representation of chromosome number 6. Chromosome number 6 contains a gene called the HFE gene that is related to iron metabolism. There is a mutation that sometimes occurs in this HFE gene that is related to the development of a disease called hereditary hemochromatosis. This mutation, this mutant allele in the HFE gene, is known as C282Y. It is listed up there at the upper left portion of the screen. And it turns out that the allele frequency for the C282Y mutation in the HFE gene is, the allele frequency is 0.223. Now, the HFE gene is located relatively close to another completely different gene known as the HLA-A gene. One of the alleles for the HLA-A gene is known as HLA-A3. The allele frequency for the HLA-A3 allele is 0.15. Now, if you could mathematically predict the frequency with which you would expect to find the C282Y mutation and the HLA-A3 allele both together on the same chromosome. We would expect that they would be randomly distributed with respect to each other, and if they were randomly distributed with respect to each other, then we could determine the frequency with which they are both found together on the same chromosome simply by multiplying together the allele frequencies for those two particular alleles. If we do that mathematically and multiply together the allele frequency for those two alleles, we would predict that they would be found together on the same chromosome 3.3% of the time if those alleles are randomly distributed in the population. But when we look at real chromosomes in the real population, we find that they actually are found together. Those two alleles are actually found together on the same chromosome much more frequently than that. That is, mutant allele C282Y and allele HLA-A3 are actually found together 15.6% of the time on the same chromosome. That is, they are found together about 5 times as frequently as you would predict by chance alone. We call this increase in the amount of time that they are found together linkage disequilibrium. Linkage disequilibrium. It simply means that those two alleles are not randomly distributed in all the chromosomes of the world, and they are non-randomly distributed. That non-random distribution is known as linkage disequilibrium. The basic idea here is probably, or is, that the original person who, in which the C282Y mutation occurred probably also had that HLA-A3 allele. And the idea is that as those alleles were inherited down through many generations, there is an opportunity for crossing over to occur between those two genes to eventually make them randomly distributed in the population. But the idea is that those two genes are so close together that it will take many, many generations in order to get them randomly distributed. And so there simply have not been enough generations of humans yet to give a random distribution between those two genes. That is why we find linkage disequilibrium. The simple fact that we find linkage disequilibrium tells us these two genes are close together. This diagram illustrates the relationship between linkage disequilibrium and the recombination frequency between two genes. And what this graph basically says is that if the recombination frequency between two genes is very small, then it will take many, many, many generations in order to get those genes randomly distributed with respect to each other. That is, linkage disequilibrium will persist for many, many generations. However, if the two genes are further apart, that is, if recombination frequency is larger, then it will not take very many generations to get them randomly distributed, and the linkage disequilibrium will not persist for very many generations. The next diagram is an illustration of what has been found in a relationship between linkage disequilibrium and the distance between genes for a number of known DNA markers. And the basic idea here is that if two genes are very close together, you are very likely to find linkage disequilibrium. If two genes are further apart, you are not likely to find linkage disequilibrium. So the bottom line conclusion is, if you find linkage disequilibrium, two genes are linked, and the greater the linkage disequilibrium, the closer together the two genes are. Now, one of the uses for the data that we've collected by gene mapping is in the cloning of genes. When we are studying a genetic disease, we often want to clone the gene that is related to that genetic disease in order to study its DNA-based sequence, to find out what that gene does and how it works. Through such studies, we can determine how mutations have affected that gene, and maybe we can figure out how to cure the problem, how to fix the problem. This screen shows us the basic approach to the cloning of genes, making use of the DNA markers and the gene mapping information that we have developed through the techniques discussed in this chapter. If we want to clone a gene, one of the first things that we have to do is find linkage to a known marker. That is, we have to identify one of the DNA markers that is located close by the gene related to the genetic disease we are studying. Once we have identified a marker close by that gene, we study that DNA region to identify protein coding regions or protein coding genes in the area of that marker. These protein coding genes are possibilities for the identity of the gene related to our disease. And then finally, what we have to do is to compare the DNA sequences in those protein coding genes between affected and normal individuals. What we are looking for is mutations in the DNA sequence of some gene in affected or mutant individuals that will then identify that particular gene as being the one that is mutated in this disease. An illustration of this basic idea is shown in the next series of slides. Suppose we wanted to identify the particular gene that is associated with some genetic disease, and we want to clone that gene. When we start our project, we are likely to have no idea where the gene is located. We have no idea which chromosome it is found on. And so we might draw diagrams that illustrate all the possible chromosomes. I'm only showing 4 chromosomes in this slide just to simplify things. We have no idea which chromosome this gene is found on. However, what we do know is the location of many, many DNA markers. That is, as a result of the Human Genome Project, we know the location of many RFLPs, VNTRs, STRs, and SNPs scattered all over all the chromosomes. And we can use all those DNA markers as mile markers to determine where we are as we're studying each chromosome. What we have to do now is to identify one of these markers that is genetically linked or that lies close by the gene related to the particular disease that we're studying. Basically, what we have to do is to repeat the experiment that we looked at a few minutes ago, in which we followed inheritance of the neurofibromatosis gene and RFLP gene in a particular family. We have to do that kind of experiment many, many times following inheritance of our particular disease-causing mutation and each of the many different DNA markers scattered over all the chromosomes. For most of those DNA markers, we will find 50% recombination between the DNA marker and the disease-causing mutation, indicating a large distance between the markers and that mutation. However, eventually, by trial and error, we are likely to eventually find a particular DNA marker that gives us less than 50% recombination with the disease-causing gene. The particular DNA marker in this example linked to the disease-causing gene is surrounded by that box in this diagram. Perhaps with this marker we found 10 or 15% recombination between that marker and the disease-causing gene. That tells us that the disease-causing gene is somewhere in the vicinity of that DNA marker. We still don't know exactly what the gene is, but at least we've narrowed the search down to this region of chromosome number 10. Now, the next thing that we need to do is to try and identify protein coding genes in that region to identify candidate genes, that is, those that may be the gene related to our particular disease. As a result of the Human Genome Project, we are very likely to know the entire DNA-based sequence inside that red box. And using computers, we can scan that DNA sequence to determine the identity of protein coding genes in that region. Our computers do that for us. Basically, what a computer will do as it is looking for a protein coding gene is that the computer will look for characteristics or structural features of protein coding genes. The computer will look, for example, for promoter regions. It will look for Tata boxes, cat boxes, GC islands located in the promoter region of genes. The computer will look for start codons and stop codons. The computer will look for exons and introns in these genes. All these are features of protein coding genes. And perhaps our computer will find 2 protein coding genes in this region. You may remember that in an earlier segment we said that almost always when a mutation occurs to cause a genetic disease, that mutation is in a protein coding gene, so that these 2 blue boxes on the screen represent our 2 candidate genes for this disease. What we have to do next is to compare the DNA sequence in these 2 genes between normal and affected individuals, and the idea is that if one of these particular genes is the gene related to this disease, then we should find mutations in that gene in the affected individuals. That will indicate that that gene is the disease-causing gene, and once we have identified that gene, then we can use our recombinant DNA technology to go in and clone that piece of DNA out of chromosome 10, isolate that piece of DNA in a plasmid to produce lots of copies of that particular gene, so that we can then study the structure of that gene in the laboratory, determine how the mutations have affected the structure of the gene, and perhaps how we can fix the problems caused by those mutations. So, in this chapter, we have looked at techniques for gene mapping. We have seen how we can use recombination frequency and linkage disequilibrium to identify the location of genes and the distance between genes, and we have seen how we can use some of that gene mapping information to help us to clone disease-causing genes. In the next chapter, we will see how we can also use this gene mapping information for genetic diagnostic purposes, for genetic testing of families in which mutations are being inherited. We're continuing our discussion of medical genetics. I'm Dr. Vernon Reichenbaker from Marshall University, and in this segment we will be covering Chapter 6 in your book, Genetic Diagnosis or Genetic Testing. Genetic diagnosis is an important development that has evolved or come out of our ability to map genes, as we saw back in Chapter 5. In genetic diagnosis, we have developed laboratory techniques that will allow us to identify which individuals carry mutant alleles related to particular diseases, so that we no longer have to rely on probability and statistics to make our recurrence risk predictions for the inheritance of genetic diseases. Using genetic diagnosis techniques, DNA testing, we can often determine exactly whether or not an individual has inherited a disease-causing mutation. In your book, you will find that there are 2 major kinds of genetic diagnosis or genetic testing. These 2 types are known as a direct diagnosis and indirect diagnosis. My experience has been that most often when students have trouble with genetic diagnostic test questions, it is almost always the indirect diagnosis questions that cause the problems. And I believe that is because a lot of people just don't understand that there really are these 2 different types, direct diagnosis and indirect diagnosis. What does that mean? The line up here in the screen could represent a chromosome or a DNA molecule. And somewhere on that chromosome or DNA molecule, there is a mutation that causes some genetic disease. If we know exactly what that mutation is and exactly where that mutation is, then we can develop a direct genetic diagnosis technique. That is, we can develop some sort of laboratory technique that will allow us to look exactly at the site of that mutation. We will examine the mutation itself. However, we do not always know the exact location and the exact nature of the mutation. And if we do not know exactly what or where that mutation is, then we have no way of looking directly at the mutation, and we must rely on indirect diagnosis. To perform indirect diagnosis, what we actually detect in the laboratory is a marker gene, one of those DNA markers that we talked about back in Chapter 5, a DNA marker that is closely linked genetically to the site of the mutation. That is, we are not able to look directly at the mutation itself, but we can detect and measure a gene that lies close by the site of the mutation. And the idea here is that if we can identify in a particular family the linkage, close linkage between a particular allele of a DNA marker and a disease-causing mutation, we will assume that since these two genes are very close together, that by following inheritance of the DNA marker, we will also be able to follow inheritance of the mutation. Because they are so close together, there is essentially 0% recombination between them, so that we will assume that the marker gene and the mutation will always travel together when they are inherited from a parent to a child. This is known as indirect diagnosis because we are not looking at the mutation itself, but we are actually detecting and measuring a completely different gene from the mutation. What kinds of tests do we use for direct and indirect testing? On the left-hand side of your screen are a list of some techniques used for direct testing. ASO probes, that stands for allele-specific oligonucleotide probes. Allele-specific oligonucleotide probes are artificial probes that we can make in the laboratory using our gene machines or oligonucleotide synthesizers. That is, if we know the exact base sequence of a mutation and the sequence of the normal allele for a particular gene, in the laboratory we can make a pair of single-stranded DNA molecules, one that has a base sequence that corresponds to the sequence of the normal allele, and a different one that has the base sequence corresponding to the mutation. We can then probe our patient's DNA with that pair of single-stranded DNA probes and determine which of those probes can base pair with our patient's DNA. And that, using such ASO probes, we can determine whether or not a person has inherited normal or mutant alleles for a particular gene. Another technique that we can often use for direct diagnosis is PCR, the polymerase chain reaction. If we know exactly what that mutation is and exactly where the mutation is, we can amplify the DNA sequence surrounding that mutation to determine whether a person has inherited the normal or mutant allele. An example would be the most common mutation that causes cystic fibrosis. The most common mutation causing cystic fibrosis is a 3-base deletion, so that the normal allele for cystic fibrosis is 3 bases longer than the mutant allele. By by amplifying that segment of DNA using PCR and measuring the size of the PCR product, we can determine whether an individual has inherited the longer normal allele or the shorter mutant allele. Another technique that we sometimes use is DNA sequencing. That simply means determining the complete DNA sequence in the region of the mutation. We only do this one if we have to. That is, DNA sequencing is relatively expensive and time-consuming, so we will typically only do DNA sequencing if we have no other way of detecting that mutation. Very new kind of direct test that is often done is a kind of test called a DNA chip or a gene chip. This is an example of microarray analysis, and this is another technique that has arisen from the Human Genome Project and all we have learned about the structure of DNA and the technology that has been developed for manipulating DNA. A DNA chip is a small little chip that is basically about the size of a microscope slide. Now, from our Human Genome Project, we now know the DNA-based sequence of many normal genes as well as many mutations causing genetic diseases. And with the technology that we have developed, we can now go to the laboratory and synthesize single-stranded pieces of DNA that correspond to all of those known normal and mutant DNA sequences. And with our technology, we can put tiny little spots of each of those manufactured pieces of DNA on a small DNA chip the size of a microscope slide, so that on one DNA chip we can put, for example, 1,000 different spots of DNA representing many different normal alleles and many different mutant alleles. Essentially then we denature and incubate our patient's DNA with that chip and determine which of the spots of DNA on that chip our patient's DNA was able to base pair or hybridize with. That then, in one test, gives us a measure of whether or not a patient has inherited normal or mutant alleles for 1,000 genes at once. This can give us a tremendous amount of genetic information about our patient that will allow us to give excellent genetic counseling to such patients. We do have to be kind of careful about this genetic information, however, to make sure that it is always used for wise and useful purposes and not harmful purposes. For example, there have been some instances in which health insurance companies have canceled a patient's health insurance when they have found out the results of genetic testing. So we have to be responsible in who receives the information from such tests and how that information is used. Another kind of test that can be used for direct testing sometimes is RFLP analysis. If the mutation has directly altered the way a molecule of DNA is cut by a restriction enzyme, then we can use RFLPs for direct testing. For indirect testing, on the right-hand side of your screen, we most commonly use all those DNA markers that we met back in Chapter 5. That is, RFLPs, VNTRs, STRs, and SNPs. When tightly linked to a mutation, all of these kinds of DNA markers can be used for indirect testing. Let's look at some particular examples of DNA testing. Let's first look at some examples of direct genetic diagnosis. The first technique that's described in the first example here involves gel electrophoresis of PCR products. In this particular example, we have a patient who is suspected of having Huntington's disease. In order to confirm whether or not we've made the correct diagnosis, what we want to do is determine whether the person has the mutation that ordinarily causes Huntington's disease. And so in order to do that, what we will do is to amplify the portion of that gene that might have the trinucleotide repeat expansion that causes Huntington's disease. We will amplify that portion of a gene by PCR and measure the size of the PCR products by gel electrophoresis. The idea is that since Huntington's disease is caused by a trinucleotide repeat expansion, that gene should be bigger if this patient actually does have Huntington's disease. Here are the results that we might find. In this slide, we see the PCR products from the patient on the left and from a normal control individual on the right. Any band that is close to 100 base pairs in size will represent a normal allele. And so we can see that the control normal individual has two bands of 95 and 101 base pairs. This represents normal alleles in that normal individual. The patient has an allele of about, near the bottom of the screen, of about 104 base pairs. That would represent a normal allele also. However, we can also see that this patient has a band that's much higher, much larger in size, of 170 base pairs. This would be the band that indicates that this patient has shown trinucleotide repeat expansion in this gene, and it would confirm that this patient actually does have Huntington's disease. The second example involves the use of ASO probes, or allele-specific oligonucleotide probes, for direct genetic analysis. In this particular example, we are testing a patient for hereditary hemochromatosis. Now, the idea is that we know the most common mutation causing hereditary hemochromatosis, and because we know the DNA base sequence of both the normal and the mutant alleles, we can, in the laboratory, prepare two small single-stranded pieces of DNA that we can use as probes. We can make one of these ASO probes that is so specific that it will only be able to base pair with the normal allele, and we can make another ASO probe that has a base sequence so specific it will only be able to base pair with the mutant allele. We amplify our patient's DNA with using PCR, the polymerase chain reaction, and then probe that patient's DNA with our pair of ASO probes and determine which of those probes our patient's DNA can base pair with. This is what an ASO probe result looks like. The numbers 1, 2, and 3 refer to 3 different individuals whose DNA we are testing, and we take DNA from each of those 3 patients and put 2 spots of DNA from each patient on the surface of a special filter. We then will probe that top row of spots with our normal ASO probe. That is, this is a probe that is so specific it will only be able to base pair with the normal allele for this disease. And the bottom row of spots will be probed with the C282Y ASO probe. This is a probe that is so specific that it will only be able to base pair with the mutant allele for hemochromatosis. If the spot is empty, that means that the probe was not able to base pair with that patient's DNA. If the spot is filled in, then that means that the probe does base pair with that particular sample. So that we can see that for patient number 1, there is a very dark spot only in response to the normal probe. There is no spot in response to the mutant probe. So patient number 1 would be homozygous normal. He has only 2 normal alleles. Patient number 2 does react with both probes. we can see that the spot here is drawn as gray or intermediate in darkness. That is because he has one copy of the normal allele and one copy of the mutant allele. Both of those probes can react with his DNA. Since he or she gives a positive reaction with both probes, that means that patient number 2 is heterozygous or a carrier for hemochromatosis. Patient number 3 gives a strong reaction only with the mutant probe and no reaction with the normal probe. That indicates that patient number 3 is homozygous mutant, or does carry both copies of the mutant allele for hemochromatosis. Another technique that can be used for direct testing is RFLP analysis of PCR products, restriction fragment length polymorphism analysis of polymerase chain reaction products. In this example, we're going to be testing a patient for Gaucher disease. Gaucher disease is an autosomal recessive disorder. What we will do in this example is amplify the patient's DNA using PCR to produce a particular size of PCR product, and then we will cut that PCR product with a restriction endonuclease. The difference between the normal sequence and the mutant sequence is that the normal sequence for this gene cannot be cut by this restriction endonuclease. However, the mutation causing Gaucher disease has caused a change in the way the sequence can be cut with the restriction enzyme, so that the mutant sequence will be cut into 2 smaller pieces. So that when we look at the sizes of these pieces on a gel, we will find that if we find the larger DNA band, that is the one near the top of the gel, the one of 160 base pairs in size, that band will represent the normal allele. The pair of smaller bands represents the cut piece of DNA that we would find if the mutation is present. In this particular example, the mother and the father are both carriers for Gaucher's disease because both the mother and the father have the upper normal band and the pair of smaller bands representing the mutant allele. The child that is listed as affected female is their daughter that does have Gaucher's disease, and we can see that she only has the 2 smaller bands, indicating that both copies of her gene are mutant, are represented by the mutant alleles. Notice that her bands are drawn darker than the bands in the parents. That's because she has 2 copies of the mutant allele, whereas each parent has only 1 copy of the mutation. Sibling number 1 has only a dark band at the higher molecular weight. That indicates that sibling number 1 has only normal alleles, has 2 copies of the normal allele. So sibling number 1 will be homozygous normal. Sibling number 2 has the 2 dark lower, smaller molecular weight bands indicating 2 copies of the mutation. So sibling number 2 will also have Gaucher's disease, just like the affected female, the one that is labeled affected female. So in this way, we can identify people that are homozygous normal, heterozygous carriers, or people that are affected with this disease. Another kind of test we can do involves RFLP diagnosis for a disease called myotonic dystrophy. Myotonic dystrophy is another one of those diseases caused by trinucleotide repeat expansions. And what we can do is look at the DNA, or look at the genes by the technique called Southern blotting. And as a result of the trinucleotide repeat expansions, the mutation will get bigger as we go down through the generations in a pedigree. Remember that the trinucleotide repeat expansions lead to anticipation, the fact that as we go down through the generations of a family in which such a disease is inherited, the mutation gets worse and worse, the trinucleotide repeat expansion gets bigger and bigger, and the disease expression gets worse and worse as we come down through the generations of a family. This diagram shows a pedigree of a family in which myotonic dystrophy is being inherited, and at the bottom of the figure is shown southern blots of DNA from each member of the family. In this particular diagram, any band of DNA that is either 9 or 10 kilobase pairs in size near the bottom of the southern blot, any of those bands are normal. Any band of 9 or 10 kb in size is normal. However, any band that is bigger than that represents a mutation in which the trinucleotide repeat expansion has occurred. If we look in the affected grandfather's DNA, that's the second lane in the southern blot, we can see that he does have a band on the southern blot that is bigger than 10 kb. That is, it's probably about 11 kb in size. That means that indicates that he has had the trinucleotide repeat expansion. That is his mutation that caused the symptoms of his disease. When he passed that mutation along to his daughter in the second generation, you should be able to see on the southern blot that mutant band of DNA moved even further up on the southern blot. That is, the mutation got bigger, the trinucleotide repeat expanded, the piece of DNA got bigger and is probably of about 12 or 13 kb in size. When that affected female in the second generation passed the mutation along to her affected son in the most recent, the third generation, you can see that the band moved even further up the southern blot, indicating that the mutation got even worse when it was passed from that mother to that son. That is, the trinucleotide repeat expanded even more, the mutation got worse, and the symptoms probably became even more severe in that most recent generation. Those were all examples of direct genetic testing. Now let's look at some examples of indirect genetic diagnosis, in which we will not be looking directly at the site of the mutation, but we will be looking at a DNA marker located nearby the site of the mutation. In the first example of indirect testing here, we will be performing our indirect testing using STR, short tandem repeated region. We're going to be doing indirect testing for Marfan syndrome in this example using such STRs. So we'll be looking at the alleles for an STR marker located nearby the Marfan syndrome gene. So that in this example, we will follow in a pedigree the inheritance of the Marfan syndrome alleles, normal and mutant, and the alleles for the particular RFLP, that is, the screen should say STR, alleles in this particular family. And we are trying to determine which allele of the STR is located nearby, is genetically linked to the Marfan syndrome, the mutant Marfan syndrome gene. Here's the pedigree of the family. Remember that Marfan syndrome is an autosomal dominant disease, and so we will be able to follow inheritance of the normal and mutant Marfan syndrome alleles simply by whether or not an individual is affected by this disease. The numbers underneath each symbol in this pedigree indicate the genotypes of each individual for the STR that we are studying in this particular family. This particular STR is highly polymorphic, such that in the population there are 4 different alleles for this STR, giving 4 different sizes of PCR products that we would detect in the laboratory. Now, in this particular family, what you should do is look through the family members and try to determine which allele of the STR is associated with the mutant Marfan syndrome allele. And if you carefully look through all the members of this family, you should be able to tell that allele number 3 of the STR is the one that is tightly linked to the mutant Marfan syndrome allele. That, if you carefully look at this pedigree, you will see every time you find a person who has the disease, they will have the allele number 3 of the STR. So one potential test question you might see from this would be simply to identify which marker allele, which STR allele, is tightly linked to the mutant Marfan syndrome gene. Another way of asking basically the same question would be to say that the woman in generation 2 is pregnant again, and we could draw a little diamond shape on the pedigree in generation 3 representing her fetus, and we would want to determine whether or not that fetus is destined to inherit Marfan syndrome. We can do genetic testing on a fetus by either amniocentesis or chorionic villus sampling, and the idea would be that if we find allele number 3 of the marker in that fetus, we would conclude that yes, that fetus is destined to develop Marfan syndrome. If the fetus does not inherit marker number 3, our conclusion would be no, the fetus is destined to be normal and will not have this disease. Another kind of indirect testing that we can perform uses RFLPs, restriction fragment length polymorphisms, to do our testing. In this particular example, we're going to be testing for the normal and mutant alleles for a disease called long-chain acyl-CoA dehydrogenase deficiency, or LCAD. Here are the results from a particular family in which LCAD is being inherited. This set of parents has already had a child who has died from this disease, and these parents have a daughter who has been identified as being homozygous normal. The mother is pregnant again, and they have done genetic testing on the fetus and want to know whether or not the fetus is destined to have the LCAD disease. Now, since this set of parents has already had a child with this autosomal recessive disorder, we know that both the father and the mother are heterozygous carriers for the disease. That is, both the father and the mother have both a normal and a mutant allele for LCAD. And when we look at the RFLP bands, the DNA bands on a southern blot for both the father and the mother, we see that both the father and the mother have 2 bands. One of those bands represents normal and one represents the mutant allele in each parent. How do we determine which of those bands represents normal and which represents mutant? In this case, we can make the determination because we are told that their daughter is homozygous normal. When we look at the daughter's DNA, we can see that she has only the upper DNA band, and notice that that band is drawn very darkly, indicating that she has 2 copies of that upper band. The fact that she is homozygous normal tells us that she has inherited the normal allele from each of her parents, and it identifies the upper band in both the father and the mother as being tightly linked to the normal allele. When DNA testing is done on the fetus, we find only the bottom band. Since in both the father and the mother the upper band corresponds to the normal allele, that means in the father and the mother the bottom band represents the mutant allele. Since the fetus has inherited 2 copies of the bottom band, that means that the fetus has inherited 2 copies of the mutant allele from the parents. So this fetus is homozygous mutant. This fetus is destined to have LCAD disease when it is born. In this chapter, we've looked at genetic testing, genetic diagnosis. We've looked at examples of direct diagnosis and indirect diagnosis. And these kinds of tests have been very useful in medical genetics for some of the reasons that are shown on this screen. Using such genetic testing, we are able to do carrier detection for recessive diseases. We are able to do diagnostic tests for late onset diseases, such as Huntington's disease, so that, for example, we don't have to wait until a person is 40 years old to determine whether they have inherited Huntington's disease. We can do the diagnosis at a much earlier age. We can also diagnose or detect the presence of mutations in those diseases that show incomplete penetrance. We can do prenatal diagnosis for these mutations using either amniocentesis or chorionic villus sampling. And we can also do pre-implantation diagnosis. That means if a couple suspects that they are carriers for mutations for some genetic disease, we can do in vitro fertilization. That is, we can fertilize an egg in the test tube and allow that embryo to grow to an 8-cell stage. And at that 8-cell stage, we can remove one single cell from that early embryo and perform our genetic tests on that one single cell to determine whether or not that embryo is destined to have the disease or not. Removal of that one single cell does not hurt the embryo, and so if we determine that that embryo is normal, then it can be implanted in the mother to guarantee that those parents will have a normal child. Genetic testing has become extremely valuable to us in the clinic in allowing us to give very good genetic advice to our patients. For your exam, genetic testing is a very high-yield topic. You can absolutely be guaranteed that you will see examples of genetic testing on your exam, so you should make it your goal to study all of the examples that we saw in this chapter so that you will be well prepared for the exam.